In London, a research team at Imperial College has taken on a highly specific challenge in insect genetics to insert genes into the Y chromosome of the Mediterranean fruit fly using the CRISPR-Cas9 technique. The aim is to create a fluorescent marker that only appears in male flies, enabling early sex separation, a crucial step in genetic pest control. Led by Angela Meccariello, the group is developing a precise protocol for Y chromosome integration, which will be a critical contribution to the EU-funded REACT project, whose mission is to develop scalable, sustainable alternatives to chemical pest control, with a particular focus on the sterile insect technique.
Our research focuses to generate the genetic sexing string. So at the pupa stage, we will be able to separate male from females. To do that, we want to integrate a phenotypic marker on the Y chromosome. So females will be white color and males brown color.
To improve their chances of Y chromosome integration, researchers must hatch and inject thousands of fruit fly eggs. Serafima Davidova conducts this delicate process.
We've injected here over 30,000 eggs for the purposes of Y integration. The type of injections that we're doing for the Y are site-specific, which means that we're targeting a particular region on the Y chromosome. So what I would do normally is make a slide to add halocarbon oil, and then have my slide cover slip, stick that on. Then we need glue to stick our eggs on so that they don't slide when we do the injections, and freeing it from the chorion. So I'm taking it from here and then I'm scooping the egg and placing it on the slide.
Once the eggs have been decorionated and aligned, they must be injected with a mixture containing plasmids, the Cas9 protein and guide RNAs.
And then what we need to do is transfer our master mix, which contains the plasmids, the Cas9 protein, and the synthesized guide RNAs. I just go in and expel the master mix into the needle, hopefully without any air bubbles, and then align the center, the tip of my needle to the center, and then put my slide here. And then I just find the posterior side of the top egg. And then I return my needle. So have the egg, have the posterior. So everything goes in, but...
However, even if the injection is successful, the challenge is far from over. The Y chromosome is packed with inactive DNA regions that rarely allow inserted genes to be expressed. The researchers hope to identify a stretch of DNA that enables both integration and strong expression of the fluorescent marker gene.
So when we inject these eggs, we're hoping to inject the posterior end, which has obviously the germline cells, which will then develop into sperm or eggs, which hopefully are the transgenics themselves, right? So in the following generation, that's when we look at the GFP signal.
To test whether the GFP gene has been passed on, the flies must complete a full generation cycle. Jonathan Mann oversees this cycle.
So to collect the eggs, I just have a brush them down, brush the cage, take the eggs. I align the eggs by just gently shaking them so they coalesce in the middle. I then take the eggs and equally spread them around. In a few days you should have larvae, but they will spring as well, if you see. And after 4 days we transfer them to these trays so they have more airflow. And then on day 11 we'll sieve the larvae, put them in a petri dish, and in a few days you should have adults. And then we set up a cage for that, and the life cycle starts again.
Once the G1 generation has been raised, Serafima begins screening for the expression of green fluorescent protein at the pupal stage.
Basically, these are really early pupae, and I prefer to screen for the Y integration at this stage, because that's when they are immobile because they don't have as much pigmentation, but also because we're able to see the fluorescence marker.
Even when integration into the Y chromosome has been confirmed, success is not guaranteed. The main obstacle remains expression.
The challenge is that we need to find the right region of Y chromosome, because Y chromosome consists in very condensed regions that are not expressed. So we try to modify a little bit the protocol, we try to improve the survival rates, we try to improve also the environment in the insectary to get more chances to have more fry to cross and then check the progeny if we have integration or not.
Inserting a marker into the Y chromosome of the medfly requires precision, patience, and persistence. However, each injection brings the team closer to developing this vital component of a sit-based pest control strategy.